So as we said, I'm just going to give a quick introduction. I'll do a few of the where we're at, and then Bob will follow on from me. But first of all, I'd like to acknowledge the traditional owners on the land on which we're meeting, welcome our New Zealand guests, and also acknowledge those guys, the Indigenous guys that I've had the privilege of working with over the last few years, particularly AJ and Tilly, who are sitting at that table with us today. So we've all seen this distribution map before. We know we've got one single strain here in Australia. We'd like to keep it that way. But maybe it raises a few questions as to how we'd actually respond to keep this out of the country. Do we focus on preventing and identifying those pathways that we already talked about and trying to stop the thing coming here? Would we respond if we detected it at the border? I mean, there's big questions. I know we're working on some of the interesting diagnostic capacity to identify and distinguish the different strains. But what happens if it arrived in Queensland and New South Wales, where there's already the disease here, the symptoms very similar? Would we actually respond? My thoughts are that it would be a difficult thing to actually encourage anybody to invoke a response where we're going to try and eradicate. If it turned up in the southwest of WA or in South Australia, yep, by all means we'd probably respond. So maybe we need to look at identifying what these different strains mean in terms of different host ranges, overcoming any of the resistance we might identify within species that we've already worked on. Within Australia, many of you have seen this map. Distribution along the east coast hasn't changed to any great degree. What we have seen more recently, and Mia will talk about that in the coming days, is the recent detection in the northwest of WA. So this pathogen's still moving around the country. It's a little bit slower than maybe what we anticipated. Importantly, it's not in the southwest of WA, where we have a high diversity of myrtaceae. We want to keep it out of there. The information coming from areas like Victoria, it's still not in that native ecosystem, but there is evidence that it's moving around. So it's still parks and gardens that we're identifying it. But you can see on the map here that it's sort of moving east towards that East Gippsland area. So there are more and more detections. It's quite happy in that space. Why it's not in the native ecosystems, we're not quite sure. Also in the Northern Territory, you can see it's active up there. It's quite a different climate to what we're used to in the southeast Queensland and northern New South Wales, for example. It's a dry tropics area, so a big dry period and then a lot of wet, but it's hugging the coastline there and it's ticking away in those environments. What sort of damage it's doing there, there's not really that active surveillance program, so it's a little bit hard to tell. Sorry, I keep looking to this side. So what are the impacts? I think when the disease first arrived here, based on information from Brazil, we thought, it's going to be the seedlings and the young trees that are affected. But what we're finding, it's anything from those tiny little seedlings that are just emerging to those that are starting to establish, saplings, right through to these 100-year-old trees. So nothing is safe. It's all life stages of different species. It does vary. We see in eucalypts that the younger stages, like the seedling and reshooting after fire, is most vulnerable. As they mature, they become less susceptible, but that's not the same across all the different species. So the current host range here is greater than 380 species. We're still getting changes, and we will as it continues to spread. We're getting species disappearing. So in 2013, 2014, we focused on two species, and this was a paper from Angus, who's sitting over there, where we knew these particular species were highly susceptible. So we were trying to get an understanding of what the impacts would be. So that resulted in Rhodamnia rubescens and Rhodomyrtus being listed as critically endangered, but only in New South Wales initially. It took many, many years before it was listed nationally, and I won't go into the details of why, but it's obviously a concern that there was a lack of coordination there going on. More recently, Fensham et al., based on primarily looking at rainforest species across Queensland and New South Wales, identified 16 species that are likely to go into decline or extinction within a generation. So that's quite concerning. And then there was another category Y, where there's some question over the rate of decline. There's evidence that there are species or individuals or populations that are disappearing, but it's a little bit uncertain as to whether there's resistance there or whether there's actually just slower decline rates. Interestingly, in 2012, 2013, we started looking at these species pretty much when Russ arrived. We looked at parks and gardens, we looked at nurseries, we looked in some of the native ecosystem areas. Based on symptoms alone, 12 out of those 16 species were already identified as moderately to extremely susceptible. And out of those category Y species, we already identified 15 of those species in there. But it doesn't actually trigger any response in terms of being able to list. You have to actually look at decline of species in the bush in terms of dieback. So symptoms alone didn't allow us to actually prioritise, apart from Bob pushing these species and saying, look, we really need to do something. But if we think about it, we're out now 2023 trying to collect populations or individuals from populations that are in severe decline. One, it affects the quality of being able to take cuttings. Some of these populations have disappeared completely. So imagine if we'd started actually collecting then, we would have had a lot more material we could have actually collected. Hindsight's a great thing. So we've got all those lists, but are we out of the woods? I mean, maybe we need to think about, we're not doing as much surveillance here now as what we used to. We don't have any long-term monitoring plots apart from one site. And we already know from that information that we're seeing changes. I mean, we can see in this image here, this is Syzygium corionanthum, which is a sour cherry, where the picture on the right there, on my right, sorry, in 2016, we thought this was actually quite resistant. There were other individuals within that site were in severe decline, but this one made the odd little spot, so it was quite tolerant or resistant. But 2018, that's what this tree looked like. And I know this tree is now dead. So what's happening there? Is it just that the environmental conditions shouldn't have been, because there are other trees there that were getting infected at the same time, only less than 100 metres away? Is it the pathogen? Is there a change in the pathogen? We don't know. We don't have enough monitoring plots or programs in place to really understand that. So we are seeing plant communities changing, and we haven't invested a lot of space in looking at the broader ecological consequences of losing these species, or losing multiple species within an individual site. In this particular site, it's a wet sclerophyll site, the Tallebudgera Valley, so there's subtropical environment, where all the Myrtaceae in the understory and midstory have been replaced by non-Myrtaceae. So you can look at the graph there and the decline of 3 major species that exist within that mid and understory, and that's from 2016 to 18, and all 3 species are now pretty much dead within that site. We don't have those species within those sites. More recently, we did some random surveys across there, and we estimated that there was about 3,400 dead or dying trees per hectare within that site. So roughly about 70,000 trees across just that site. Across that whole valley ecosystem, we're getting similar deaths and declines. So 91% of the understory and midstory Myrtaceae are dead or dying. So what changes are happening within that ecosystem? So yes, the forest is still alive, it's still a forest there, but it just doesn't have those Myrtaceae present. So in 2019, 2020, we had a lot of wildfire issues, big areas of land killed off, just severe wildfires just ripping through the country. So everyone thought, oh, well, what's it going to do to rust? So we started looking, and within probably a couple of months of the fires and that regeneration starting, rust started to appear and just started taking off, because everything was reflushing, perfect conditions for rust to develop. And these are 2 species, just as an example, in the coastal heath areas, 2 paperbark species, Melaleuca nodosa, which is the prickly Melaleuca, and Melaleuca quinquenervia, which is one of the broadleafed paperbarks. So in that, you can see on the graphs there that over time, these are short-term monitoring plots. We stopped these after about 18 months. That in that, the Melaleuca nodosa, we had close to 50% of the trees dead, with the rest in severe decline. As you can see there, the numbers of trees that flowered, only 6% that were producing flower. So that's reducing that. Yes, there might be selection for resistance of 1 or 2 individuals, but do we really understand the genetics of the host in terms of being able to understand whether that's going to be enough for natural regeneration or natural selection of resistance to occur, or do we need to step in and actually assist? So Melaleuca quinquenervia, similar declines, and this was mature trees. These are big trees that were killed, and all the reshoot, reinfection, we're getting over 60% of trees dead within that 18-month period. There are other factors with Melaleuca. I don't think it's just myrtle rust, but there's insect attack on that reshoot as well, so it's a combination of rust and other things occurring. Flowering on 16%. Yeah, great, that might be great. We do have resistance potentially within those populations. This is just to show what it looks like in the reshoots that we get after fire. So that's Melaleuca nodosa on the left there, and the other two are on the Melaleuca quinquenervia. So it basically looks like somebody sprayed the trees with chemical with that burn occurring on those new shoots. And repeated infection, they just run out of reserves and die. So what about seedling recruitment? So because of issues with COVID, border access and that sort of thing to get to these sites, we would have loved to have done these more frequently, but basically a lot of the surveys for seedlings were one-off assessments. It should have been monitoring plots, but I had to go through all sorts of levels of government to actually get permission to go across the border to get these, and then the border actually shut. So, but interestingly, at the time, and I'll say they're the resistant or they're disease-free, in 2 of those sites we had around 30% resistance within populations, or disease freedom within populations. That matched some of the glasshouse studies that we did earlier on with a lot of the New South Wales population, so that was interesting. But as you can see from the images there, that timing is interesting. You can get, obviously, infection when they're tiny, but what we were seeing when we were coming back to these sites, that quite well-established seedlings were only just starting to get infected. So that single one-off assessment is maybe not giving you an accurate indication of what's actually going on. On the other coast, there's Chatsworth, which is quite an isolated population. It's well away from those normal structures. It's an inland wetland area, and there was only 15% of individuals showing disease tolerance within that site. Tilly and AJ are going to talk more from this space, but really in Australia we've only scratched the surface in relation to cultural significance of myrtaceae and knowledge in relation to how we might better manage this disease. We're working with different groups now to try and build a network, to try and get a better understanding in different ecosystems as to what's going on and what actually can be put in place to protect country. Screening for resistance, basically it's been focused primarily on commercial species because we did have a lot of collection material, broad range of populations, so it's much easier to look at resistance patterns. So it was focused on, obviously, eucalyptus and corymbia, which are key forestry species. Lemon myrtle, we were lucky enough that somebody had actually done a collection of all the germplasm prior to rust, not for the purpose of rust, but looking at the commercial viability of the different provenances. And then tea tree. And then there are, I know Perry's done some work with Chamilorcium, the gelatin wax in WA, but otherwise environmental. We're looking at paperbark, the Melaleuca species, and that's about it. So maybe posing some questions. So have we learnt the lessons? Are we better placed from a biosecurity perspective? I'd say yes, we are a little bit better placed because we do have a department that's focused on environment now. But how does that sit in relation to industry? I think industry is still up here, environment's down here in terms of a voice when it comes to that biosecurity space, particularly incursions. Is there an appropriate Indigenous voice, and how do we actually do that? How do we actually identify the groups and have everyone have a voice from an Indigenous perspective? I don't have the answers. Is the message getting out there? Well, maybe not. We talked about those pathways. Yes, we can identify those official pathways, but it's probably the highest risk ones, those pathways that we can't control. And this was an article out of the ABC just recently, where you can see a whole range of different things have come into the country where people have tried to hide them. So this is stuff that they found. And so you can imagine if there's all these people they did detect, what's come in that they haven't detected. And you can notice on here that someone brought in macadamia plants. There are 4 species of macadamia that are already listed as endangered in Australia. So this is a massive problem. So where do we invest? Do we invest in trying to identify that, those particular strains in South America, they're a risk to our eucalypts, or maybe they're not, because this is a pathway we can't control. How can we get the message out there? What are our target groups? So to stop those sort of illegal imports, is it that we just need to educate people about the consequences of these things arriving in Australia? Do we focus on the next generation? And what sort of messaging will work? And we put out the video, the myrtle rust silent killer, and great, it got a lot of hits and that sort of thing, but did it reach the right people in terms of getting a response that will result in actions, publications, presentations, articles? There's all sorts of things, but we need to have a really good conversation about what direction we're going to actually take. Thank you, and I'll hand over to Bob.